This challenge is about the design of a miniature head-mounted optical system for wireless photometry in mice. My name is Karthik, I'm with the University of Calgary, and my group designs electrical and optical instrumentation, primarily for basic neuroscience research. A little bit about where this is coming from. Now, a lot of what we know about the brain, how it works normally, and what goes wrong in diseases has come from investigative tools which were applied in animal models. Fiber photometry is one such tool, and the idea there is an optical fiber delivers light to the brain. It also collects light from the brain. Now, using dichroic optics, we can separate this excitation light and the signal light. And then analyzing the signal light can tell us something about what's going on in the brain near the region where the fiber tip has been implanted. Here's a picture of what this is all about. You can see the fiber here, which essentially carries light both to the brain and also from the brain. And then there are benchtop optical systems which do the separation of the light, and the analysis is done. The analysis is done offline. Now, as you can imagine, such a system requires a fiber optic tether. Now that is a problem because it limits the duration and the kind of studies that can be done. For example, you couldn't have a mouse run through a maze or obstacles with this fiber optic tether because the tether would simply get caught on these on the obstacles in the arena. Another example could be a social experiment where, let's say, we have multiple mice interacting, and then if each of those had fiber optic tethers, that would clearly be a problem. Now, wireless systems do exist, but everything which is out there right now uses a lot of optical power. In the sense, you're blasting the brain with a lot of light. And the problem there is this light causes phototoxicity. It essentially damages structures in the brain. And as a result, you can't really use these systems for more than a few minutes at a time. And that again kind of precludes a lot of studies that can be done with this technology. So what we're looking for is a miniaturized system that you could mount on the head, and it would have integrated electronics and optics, so there would be no tethers required. So this challenge focuses on the optical aspects of the system. In particular, we're looking at integrating a light source, focusing optics, dichroic optics, and a photodetector. This is what a typical setup looks like. We have an optical fiber, the diameter of which is typically around 400 microns. That is housed in a ferrule. Standard diameters are 1.25 or 2.5 millimeters. So usually we have a light source, typically a light emitting diode, and then a collimating lens which collimates the light coming from the LED. Next we have an excitation filter which rejects everything except blue light, and a dichroic mirror which reflects the blue light towards the ferrule. Next we have a focusing lens which takes that light and focuses it into the optical fiber. Now when light comes back, what we have is signal light, which is shown in green here, and some of the back reflected blue light as well, which we want to reject. Now the focusing lens collimates that light towards the dichroic mirror, which this time transmits only green light. Next we have an emission filter which again selects for the green light, and finally we have another focusing lens and a photodetector which converts that light into an electrical signal. So that seems fairly standard, and indeed the technique that I just described has been used for decades for fluorescence imaging. The challenge is mice are very small. Typical lab mice are 20 to 30 grams, which requires everything that I've just described to be miniaturized into an assembly which is about a gram in weight and occupies less than a cubic centimeter. We should be able to deliver about a microwatt of blue excitation light. We need to be able to reject reflections by a factor of about 1,000,000, and we need to be able to collect at least 80% of signal light. So this challenge is going to require expertise in mechanical and optical design to fulfill the specs that we are looking for. Now, this is, as you can imagine, a research-based challenge, and what it would lead to is a publication in a scientific journal and the adoption of this technology in neuroscience labs across the world. There are companies which sell fiber photometry hardware. The problem is this is still a very niche market. I'll be supportive of any open source or commercial endeavors that any innovators are going to be interested in. So that's everything I have, and I look forward to working with some of you on this challenge. Thank you.